Well, we've begun. So, without further ado, we are starting the very last unit. I get this at home. I get ignored at home. It's okay. It's all right. It's all right. Very forgiving. Don't do this with other teachers, please. They have attitude. Did I vote? No, yesterday was voting.
No, post.
Did I post it? Anybody? Did I post it? Did I send this to you?
I don't think.
Okay, I was going to ask that question. I'll do it right this instant. We have plenty of time. You always get short shift. I'm sorry. I feel for like an inch not adequate. Just a little bit. Oh, because now you know I have to log in. Oh, please, will you make me log in? I hate that. I have to make you ill. I'm a 19 today. No, it's the wrong number. Try again. Denied. Try me again. I've been accepted. Ah, cool. Courses for horses. All right, I'll get this one to you and do the other ones, so we don't waste any more time on that. Massage, news. All course members. That's you and attack, because these are big. These files are fairly big.
That's going to be on the exam bill, right?
This is not on. This is all new material.
Okay.
Everything here. This is starting now. So everybody got that? If you're looking, okay. So I'll in between classes, I'll get the rest of it going. So apologies. There are 3 PowerPoints that are involved in this particular sequence. I will load the Welsh Hole PowerPoints. They'll be under Unit 4. I'll name it Unit 4, and I'll put whatever documents are in there other than this being sent to you, of course. This one is more about structure. There's a little bit of function in this. This is mostly structural. This is microscopically based structure for the most part, and sort of a general get-together with the nervous system. Number 1. Number 2, okay, the one that really is filled with physiology is the second PowerPoint. PowerPoint B, that's really how the nerves work. It demonstrates what I'm going to be talking about with myelin and how the signal is transmitted, why it's rapid, etc. All the physiological stuff. And it reinforces everything we really studied, the neuromuscular junction on the nerve side. And the last one is extremely important as well, because that's really where we get in depth into neurotransmitters. And that's really the jumping off point for what we're going to cover today, or now, as it were. So here we are. It looks bad. So you have 2 major control systems, the nervous system and the endocrine system. The nervous system is for right now, fast, microseconds. The endocrine system is slower, not slow. The ultra fast is nervous. The endocrine system, we're not talking about microseconds. We're talking about seconds, minutes, hours, days, maybe a week. So there's a lot of variation in endocrine, depending on how long the chemicals that they release last. But the nervous system is that's your ultra fast response system. When you're driving in the car and you got to make a sudden move to avoid being in a crash, that's your nervous system. When you're after that and and all of a sudden your heart's racing and you're, oh my God, he could have killed me and blah, 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 blah. All that stuff, that's your endocrine system. One's super fast, the other one, and if you recall when you've had something like that happen, how long does that linger? How long does your heart race? All those kinds of things. We are going to explore some of that at the very last chapter we do. And the very last chapter that we do is called the autonomic nervous system. And that's really sort of how the regular nervous system sort of makes its mark on the endocrine system, and very much as a part of homeostasis. And that's how we conclude. So now we're in the home stretch. So we have 3 major chapters. We're going to be, in addition to this using microscopic and physiology. The second portion of this trio is really looking a little bit more in depth to some of the stuff that you learned in lab with regard to the central nervous system, parts of the brain, kind of a little bit more about what they do, some special aspects of it. And then lastly, this autonomic nervous system, which undoubtedly we'll be doing on the last week. You didn't crash. Good, good. You used to have to drive your car? It sounded like, oh my goodness. Nevertheless, rapid and specific, immediate responses right away. Basically, people are immediately confused and understand that this is basic. Signals come in, we process them, and signals go out. These are called afferent or incoming. Notice I accent the word with the first letter, because if I say afferent and efferent, hard to distinguish them. Always be very clear when you're enunciating something and when you're writing something like that. So afferent or incoming signals, efferent, which derive, and both these terms derive from this, which are effector or outgoing signals. And you're going to see the terms used a lot anatomically, physiologically. Afferent and efferent play an important role, as you will learn. And the efferent structures that the nervous impulses are going to are usually muscles and glands. Vast majority of these types of things. And so it might surprise you to know that the integration, which we call interneurons or in between neurons, the vast majority are interneurons. 98, 99% of your nerves are processors. Contrary to what a lot of people think, they aren't mostly sensors and effectors. They're not. It's all about, and it's really a microcosm of what we do with things. I mean, I told you about that. Oh, but what clued you into being able to respond so quickly? You're visually taking in the picture. You're cognizant of how fast you're going. You're cognizant of the cars that are around you. All of those things. You're processing that information, and yet instantaneously you can respond. That's the beauty of the nervous system. Because you're I mean, certainly if you're going to be in a vehicle, you should be, and it doesn't surprise anybody. And when people are distracted, the whole texting thing and stuff like that, how difficult, how any distraction at all might prevent you from making that maneuver that could avoid injuring yourself, others, chain reaction, whatever. So sensory, afferent, integration, interneurons or in between neurons, and lastly, motor output or effectors. So the nervous system is, guys, zip it, please. Good Lord. God, if you use high school, you have to separate you. Goodness gracious. So 2 divisions. You should have had this in lab. I'm hopeful that you did. The central nervous system. You learned about it and where it was in that dorsal body cavity you talked about the very first day of lab. Ventral cavity, the abdomen, the diaphragm, the thoracic cavity, abdominal pelvic cavity. Those are ventral, dorsal. One thing is in there, central nervous system. Definition, brain, spinal cord. Slam dunk on the test for the final. It's a secret. Loose lips. Those are my son's a naval officer, loose lips. Very true. So integration and control center. That's what it does. And really it's receiving the data and it's acting upon the data, but it takes a lot of processing. The peripheral nervous system is anything that protrudes. And it does so by way of nerves, whether they're from the brain, cranial nerves, or from the spine or spinal cord, peripheral nerves. Okay, that's the 2 divisions. We are not going to really look at the peripheral nervous system other than some descriptions and some comparisons, which will start a little bit today. That's a typical illustration, no big deal for us. To me, that's more lab when you start talking about what nerve is what nerve, that kind of thing. You all did cranial nerves, that kind of stuff. That's really where the lab end of it comes in. When you see the term somatic, it means coming from your body. And the implication is probably the best way to describe it is anything then from the internal organs, which are visceral. So if it's coming from the skin, the joints, the muscles, tendons, anything that is giving you, it's supplying you with information about your body positioning, what's going on. Am I sitting on something? Is it pressing? Is it hot? Is there something in my pocket that's I'm sitting on that's uncomfortable? All that stuff. Those are described as somatic sensory. Visceral is internal. We don't have a lot of those comparatively. We have a lot less sensory information coming from the interior. And it's somewhat of a confusing area. I don't know if we go over it in this class or not, but we'll leave it at that for now. And the efferent or motor division is basically muscles and glands. That's the body part. When it goes to the viscera, what's known as the visceral motor division, that is the autonomic nervous system. We dedicate the last chapter and probably the last week of our class where we gather on that. There is a class Monday before finals. I'll probably make that a review session because we always end here. Since you're all exams are always Tuesday to Friday of exam week. We make up for the Monday we lose on Labor Day, and then in the spring we make up for the Monday we lose on MLK Day. Okay. With the last month, so exam week starts on the Tuesday, right? If you did not know it, that's what we do. And schools all do it differently. We've been doing it this way since I've been here. So the autonomic nervous system, and in other words, you could substitute as automatic. You don't have any control over it. So this is what we control: conscious control of muscles and signals going to muscles and related structures. Voluntary. Autonomic, visceral, all the stuff that's really about AP2, but I'm going to give you the foundation as the last nugget of this 3-chapter arc that we're going on the final. It's involuntary, and you're going to see this is where homeostasis comes from. Sympathetic, just to give you a very brief introduction, is for action division. Parasympathetic is what you're active in your bodies now, relatively speaking, at rest. And you always see this chart, not a big deal for us. That's what I've written on the board. We're going to start with the nervous system, but before we really talk, and we really won't talk a lot about nerves today, the anatomy of the nerves, and there's 3 or 4 different kinds of shapes of major nerves that you'll see, and we're really not going to spend a lot of time on those, comparatively speaking. But nerves are unique in that they have their own connective tissue support system. Everything else in your body, mostly you have connective tissue. Bones are connective tissue, muscles are a unique tissue, but they have connective tissue that surrounds them, all those epimecia, you see all those mecia and stuff like that. Just think, your liver has a capsule around it. That's connective tissue. That's all basically the same construction material. The construction material, other than the nerves themselves, which are really the source of everything, are called glial cells. They derive the word glia from glue. So they're neuroglia, or neuroglia is how it's pronounced. And that's really, when you talk about histology, that's looking at the tissue level. They're small cells. They're delicate. The nerve cells are so delicate. It's like they have these number of cells, and there's only 6. 4 of them, as I put on the board, as you're looking at it over there on the far right, the right side of the screen, okay, are found only in the central nervous system. The other 2 that either have the same or similar roles, and they're the only 2 that are found in the peripheral nervous system, but they are part of this particular lecture. You will see the names over there, okay? And then lastly, I'll talk about myelin sheath gaps and nodes as we get into that part. A little bit, sort of a brief introduction to how nerves transmit signals. So the glial cells are just that. The unique thing about nerves is it's just like muscles. They're amitotic. They don't replicate. What you see is what you're born with them. Do they change a little? They enlarge? Basically, the way our nervous system developed is making multiple connections. Look at what happens when you're born. You can't do a whole lot for yourself. Basically, you can't do anything for yourself. Someone has to care for you. And it's the development of the nervous system is where we see those landmarks that excite parents and siblings and family members and grandpas like me, okay, are all excited about it. The baby's holding his head up or her head up. She's starting to crawl. She's starting to stand. She's starting to walk. Okay, talk. All those things. Those are developmental landmarks, right? That's all about the development of the nervous system. What assists the nervous system in making that development so the nerves that are there properly connect with one another, properly form together, hold them in place, allow those signals to be transmitted, are said glial cells. Nerves are amitotic. You can't get any more nerves. Okay. The good news about that, they don't get cancer. To anticipate a question, what about brain cancer? Brain cancers are in the glial cells. People get gliomas. Another term, you'll see astrocytomas. You'll see schwannomas. Okay, most of them are benign, some are malignant. Okay, so nervous system, it's about the glial cells because they're the ones that can change and still undergo mitosis, which is not the same as the nerves themselves. So that's an important distinction. So we're really going to be looking at the transmission of the signals after we look at the anatomy. And the anatomy, I don't know that it's necessarily that intense when you just because you don't have a whole lot of cells to look at. And really, we only focus on one type here. Maybe a little different in AP2. Here are the four big players: astrocytes, microglial cells, ependymal cells, oligodenders. We'll start here. Astrocytes, they're the most of them. Astrocytes do the most roles. Astrocytes are the principal cell that's involved. One of the things, and you'll see that, abundant, versatile, branched all over the place. Astro means star. So they're star-shaped cells. A lot of your cells have that type of thing. They have tentacle-like projections. Okay. Another name, you learned about them in the skin when you looked at dendritic cells. Cells that had little tentacle-like projections that were in the base layer, but their little tentacle-like projections could sample. You're going to get to know them again when you hear about them being called antigen-presenting cells. So it's a common way that cells look. You're going to see that with white blood cells as well. Moving on. Highly branched, and they cling to the nerves. They cling around where the nerves join. Synapses, the same term. Wherever a nerve and its target be another nerve or a muscle or a gland, where they come together, very often we call it a synapse. And particularly so around capillaries, which are how nerves derive their nutrition and their oxygen supply. And that's really a pivotal role. What's very unique, again, when you say unique, it's not a little term. What's very unique, again, is the fact that something we're going to go over more than once is called the blood-brain barrier. Capillaries are like this. Capillaries are just lining cells that are held together. They're held together mostly by tight junctions. Tight means watertight. The more you have of them, the more watertight. We went over this earlier. The more you have of them, the more watertight they are. It's a structural thing. In the brain, there's so many that water and water-dissolved molecules can't pass through. That's why when someone gets medication for a nervous system disorder, if they're taking a antipsychotic, if they're taking a mild antidepressant, if they're on Prozac or Paxil or Zoloft because they have an eating disorder or OCD or whatever, okay, those are medicines that can cross the blood-brain barrier. Okay, they are by definition fat soluble. They literally move through the membrane of the capillary, the way oxygen and carbon dioxide can move. They move through the membrane system, as you will see of the astrocyte, and furthermore, then they move through the membrane and into the nerve itself. So that, and you'll see that, and that's the way that pharmaceuticals are engineered to be utilized for that. It doesn't all work that way. It takes strong pain meds. Giving somebody a shot of morphine postoperatively, okay, that's a relatively small molecule that happens to be fat soluble. The more fat soluble it is, the faster it gets into your system. Okay? That's the difference between morphine and we tack on what are called acid. It's organic chemistry, excuse me, and we make it more fat soluble and turns into heroin. Why is that more addictive? It gets in there faster. See? I mean, that's where a lot of this comes together. If you wonder about how pharmaceutical science is very interesting, many of you are going to have to endure some very difficult times with pharmacology. Okay, that's a hard course. That's why I do what I do here, in the hopes that you'll be sitting in that class and go, now I know why he opened his big mouth and told us this stuff. Got it? I'm not so think as you dumb I am. I do that all the time. My other favorite saying is like, no good deed goes unpunished. That I keep doing. Functions include support and bracing. So they hold on. They're supportive tissue, as most glial cells are. This is the exchange. I'll show you diagrammatically what it looks like. This is the maturation process. By doing the exchanges, they're controlling the chemical environment. They also, and this is where they intertwine, nerves talking to one another by way of neurotransmitters alter this environment. They change it. That's part of the maturation process and how they function, and it's part of information processing. It's very I'm explaining it to you relatively well, I think, and I'm not nearly in depth that others can explain it. It is extraordinarily complex. Extraordinary. This is the diagram. And so what you see in that particular diagram is you can see here's a capillary. Again, water can't get through this one. Almost every other capillary it can. Okay. And then on top of the tight junctions, it's surrounded by membrane. It reinforces the barrier. And now it can take this material from the barrier. Okay. It can go through. So basically it goes through this membrane, carried along through the membrane of, you can see the star-like projections or tentacles into the nerve. So let's say this is oxygen and nutrients coming in, and out here, or it could be the same place, carbon dioxide and waste products coming in and being transferred to the capillary. Capillaries are where the exchange occurs everywhere in your body, where you deliver oxygen and nutrients and pick up waste products like carbon dioxide and other waste products. These are unique capillaries. In the male, they also have a barrier called the blood testicular barrier that's similar to this. And you get to that in repro anatomy and physiology. That's the very end, most likely, of AP2. Moving on. Important cells. We only find them in the central nervous system. In microglial cells are different. You're going to see the satellite cells are sort of an oddball. I'll explain. That's why I have a question mark on it. Microglial cells. These are tiny. They have thorn-like projections. They act like scavenger cells, kind of like your white blood cells do, but they're tinier. And they might gobble up, and anybody having microorganisms in this area, you're in big trouble already. I mean, viruses and bacteria in there are bad news. People die from those things very, very quickly. So you wouldn't, even though technically that's what they do, they are more about gobbling up debris. Your nerve cells are extraordinarily active and busy. You use a ton of ATP. You're building stuff, you're assembling neurotransmitters. When we looked at the neuromuscular junction, same kind of deal here. Things diffuse away, they're gobbled up by these cells, they're moved around, that type of stuff. They're very tiny, there's not a whole lot else to say about them, but they kind of act like scavenger cells, which are similar to white blood cells. Defensive is a very good way to a single term to description. These are really fun. These are called ependymal cells. I was surprised. I asked this, when you dissected the brain, did anybody look inside the ventricles? Did you identify where the lateral and third and fourth ventricles were when you did the brain dissection? Did you look in there and find some kind of brown looking yucky flat stuff? Maybe not. I mean, you have to know what you're looking for. A lot of people some debris, I'll just scoop it out or pull it out with my forceps. Those are tweezers, the uninitiated. They're not tweezers, forceps. They're instruments, not tools. The plumber has tools. Making fun of the plumber makes more money than I do. A lot, even when I was a doctor. Should have been a plumber. Nevertheless, What happens, the ependymal cells are part of something that's in that you're going to see when we look at the brain called choroid plexus. It's the way cerebrospinal fluid is finalized. The choroid plexus is a little like nest of capillaries surrounded by ependymal cells, and ependymal cells line those ventricles, lateral, third, fourth, all shortstop. Baseball joke. Nevertheless, they're ciliated because they move the cerebrospinal fluid around. What cerebrospinal fluid is, everyone, is kind of a filtrate. It's very similar to plasma, which is the watery part of your blood with stuff dissolved in it. So it's similar but not identical. And what those capillary beds do, and even with the aid of the ependymal cells, they help to finalize that fluid that becomes cerebrospinal fluid that bathes all of our nervous system structures and is vitally important. That's the stuff we talk about down the road about lumbar punctures, basically spinal taps or pulling out spinal fluid. I heard you didn't go into spinal cord anatomy. When you were, and I got it covered 6 ways from Sunday. We'll get it done. It's okay. You can learn the difference between a lumbar puncture and an epidural. Most people are familiar with epidurals vis-a-vis pregnancy and baby deliveries, and as opposed to, which is not nearly the same as a spinal tap or lumbar puncture. Okay. Moving on. So central cavities, permeable barrier because stuff can flow in and out of it, etc. And they look like this, and it's constantly moving it around. Those fluids circulate. We'll go over the circulation and how they spread around. We make about a pint every day, and about a pint every day is sucked up by little siphons that you find in the area in the thick linings that protect your brain and your spinal cord, particularly your brain, called the dura mater. It's part of the meninges or meninges. When you hear about meningitis, okay, those liners become inflamed. It's very, very dangerous, particularly bacterial. Moving on. Now, these guys are about myelin. They're called oligodendrocytes. Myelin is basically, nerves are delicate. And when you dissected that brain, you saw a ton of white matter as well as gray matter. Remember the color difference that you saw there? White matter are transmission lines. The fact that they're myelinated makes them very, very fast. The more myelin, the thicker the myelin, the faster they are. We're going to look at that in detail. Myelin is really the plasma membrane of 2 different cells. In the central nervous system, it's oligodendrocytes. In the peripheral nervous system, it is a Schwann cell. Okay? And so basically, they wrap around the nerve fibers. And what is membrane? Proteins embedded in fats. Myelin is a very specific fatty and protein-laden substance that's an insulating layer. The same way you would take, if this, an electrical wire like this, what's around it? A rubber coat. That's the myelin. If it's damaged, if it's irregular, what happens? What's a disease that causes the body to attack the myelin that you have to be very worried about? It targets women, again, like most autoimmune diseases, 3 to 4 times as frequently as men. It ends up with multiple hard spots on myelin, and it happens in the peripheral and central nervous system. It's called multiple sclerosis. Sclerosis means hardening or hard spots. MS. We're much more common than you think. Giving you some context. Now you know what it hits. It hits these myelin sheaths. And what's interesting about the ones in the brain, they're called oligodendrocytes. It's one cell, and the axons are the thin, when you see a nerve, it's made of hundreds and hundreds of axons. The axons are threads, microscopically thin, but fairly long threads. And they are, what happens, one cell can embrace and surround as many as 60 of these because they're in the brain. In the brain, your space is limited. We don't have a lot of room. So that all of those axons, even though they are independent in a lot of ways, they have to be held closely together. That's the role of the oligodendrocytes. You don't have the space limitation for your ulnar nerve, your funny bone, your radial nerve, your sciatic nerve. Okay? They can move around. They're not held in place. Okay? So if you strike your funny bone, you're compressing it against part of your elbow. You're actually should know what you're being compressed against. The medial epicondyle of the humerus, something called the ulnar groove, bone neck. But it has a lot of space there. I mean, I can roll it all the time and move it around, not hurting it. You feel it now. Not sending stuff over there. It's a little different if you compress it against the bony area. So you can see how it packs them in very, very tightly. That's a very, so it really, its membrane is the myelin. The oddball are the satellite cells. These are the ones in the peripheral nervous system. The satellite cells are just that. They're cell bodies, and let me explain it this way. We don't use the term nucleus. I'm sure you already learned this because you learned about the red nucleus and the basal nuclei in lab, I'm sure. A nucleus is a term we use for clusters of these things. The actual nucleus of the individual nerve cells are called cell bodies. You'll see another term that they're called soma, which actually means body, or perikaryon, which means around the kernel or the core. All of those are interchangeable. So the satellite cells, we don't really know what they do. They may play a similar role to what the astrocytes do. And we don't see them anywhere but the peripheral nervous system. The Schwann cells are the companion cell in the peripheral nervous system of the oligodendrocyte. Each of these is a Schwann cell. And what you see here, these are called, if we go back and take a look, these are called sheath gaps. Now, it's one of those things that 50 years ago, they were in something entirely different. And when you see some of the animations, they're going to use this terminology. So now they're called myelin sheath gaps. And really the signal doesn't go through the myelin so much as it jumps effectively from gap to gap. That's why the nerve cells conduct it very fast. The more myelin, the faster. Thicker the myelin, the faster. And we'll get to the why about that when we look at the physiology. The old term that you should be familiar with for two reasons. It's important to know, because you'll see it in the animations, and I love to say it. They're called nodes of Ranvier. That's right, Ranvier. It's like talking in French. My middle kid, who lives now in Weirton, used to live in North Versailles. I go, "Oh, you live in Versailles at the palace?" He goes, "No, I live in North Versailles." Just saying. You guys have very little appreciation for my bad jokes. So we don't really know what these do, and they function similar. Whenever you see that on a PowerPoint, may, similar to, have been associated with, that's code for we don't know. In medicine and in science, if you know, you know. Simple declarative statements. The myelin sheaths in the peripheral nervous system are from Schwann cells. The myelin sheaths in the central nervous system are because of oligodendrocytes. Simple declarative statements. There's no room, no wiggle room. And the neural lemma, remember, like a sarcolemma is a membrane. So the neural lemma is a nerve membrane. I won't use the term, I'll say Schwann cells. So again, myelin sheaths function similarly to the oligodendrocytes. They play a pivotal role in something we do not discuss here, but you probably will, depending on who's teaching you, AP2. Nerves can regenerate, not here. If a nerve is damaged in the cell body area, it's destroyed. These can be, the little endings here, and this is most of these are called axons, as you will see next time we meet. They can literally seek each other out. Chemicals are secreted when one of these is cut or damaged. And those chemicals have a fancy name. They're called nerve growth factors. We didn't even know about them 50 years ago. And they induce the nerves to basically find other nerves that are effectively open and secreting the factor. And the factor causes them to grow and grow together, albeit slowly. I had patients, you inevitably cut nerves when you operate on somebody. The biggest complaints I had, even though the surgery went great, was I can't feel this and the skin's numb. I don't like that. They weren't mad about the surgery, and they weren't it wasn't never ending. And I said, yeah, you try different massage, different types of things. There's some topical meds that work. And a year later, they came back, many of them, and go, it's a miracle that came back. And it's not. It's because nerves, we didn't know how it worked now. That's just some context. They go over it a little bit more in detail in the way that it's formed, if they do that. It's the reason why if we hear about a case where somebody was in a terrible industrial accident or a vehicle accident, they got cut and their limb, part of their limb was severed, they can reimplant it. That's because of, you can sew blood vessels together, you can get the nerves to grow together, etc., etc. There's nothing magical about that. If you just handle it properly and you get it fast enough before it deteriorates, typically there's a very good chance that you can make that work. We had that at our hospital with, if it was an upper extremity, we had a neighboring hospital had, somebody who's their hand surgeon was, one of the top guys in the city of Philadelphia. You call them if we see this happen, and we get the patient over their hospital. Now it's part of Drexel University's medical school, that hospital. Moving on. So the nerves themselves are the structural unit. They're very, very specialized, lasting your lifetime. This is the whole idea of all they do is undergo mitosis. This is important. They all have a cell body and they have processes. This is something that's really important to understand. And it's not anything I'll ask you about in this class. I don't know who's going to be teaching AP2. I'm pretty sure Dr. Maurer is going to be. Did you guys, some of you register yet for AP2? Yeah. Did they have a name? Anybody? Or was it? Do you know who was staffing it?
The lab is Dr. Robbins.
Dr. Robbins got all the labs.
I don't know.
Well, he's teaching one of them, and they don't have teachers.
Okay, because he's got other stuff he has to teach from his epidemiology, I would think.
Yeah.
Okay, and I'm sure Dr. Maurer, who comes in after me here, and is teaching micro here, is probably one of the ones. He'll probably only do one section, I would guess. Typically, that's what he does. He does one AP2, unless we need somebody to do more. But nevertheless, the point I'm making is if there's one disease, if you're going into working with people in a healthcare environment of any type, the disease you are going to see the most of if you're working with a typical adult population, with the exception of everybody has arthritis, which they do as they get older. Diabetes. You cannot possibly know enough about diabetes. It's called diabetes mellitus. That's sugar diabetes. It's not the only one. Diabetes means the great thirst. What it is about is not just high blood sugar. It's about irregularities in sugar metabolism, having difficulty using sugar appropriately, typically characterized by high blood sugar. Short definition, comparatively. If so, you turns around and tell me if you have really high blood sugar, you can die. That's absolutely true. Okay, if it's consistently high and it isn't corrected, you're going to die slowly. What this happens, and if you know a diabetic and they're not careful, particularly an insulin-dependent diabetic, this can happen to them. I had many patients, unfortunately, and arguably my favorite patient of all time, who died some years ago, unfortunately, who was a very, very brittle insulin-dependent diabetic. Low blood sugar can immediately lead to your death. High blood sugar is slow. Emergency is low blood sugar. Your brain can only exist on glucose and glucose alone. No other sugar will do. And it must have oxygen. Without those two things, it dies. And once it dies, again, you can't make more nerve cells. This is what happened to a lot of them. This is exactly what happened to him. He's in a hurry. He took his insulin first thing in the morning. You're instructed to eat something. Okay. I'm late to work. Hopped in his car. He sat there telling me the story because I heard he was in the hospital. The next time he came into my office, what happened? He goes, I don't know. I remember parking my car at the parking lot at work. He works up, right? If you know where Titusville is, right? Titusville. And next thing I know, I was in the hospital, waking up. And it's like in the movies. They give them a shot of sugar and all of a sudden it's like, where am I? I feel fine. What am I doing here? It's like it corrects everything instantaneously if you get it in time. It's a hypo, which means too low, glycemic crisis. He is fortunate because a lot of times what will happen, the best outcome if you happen, it would be just you running into a tree with your car and hopefully surviving, or like that, the parking lot. The worst is you black out and your car is moving. You could hurt, you could kill other people as well as yourself. It could be extraordinarily tragic. That's why it's so important. He did the dumb thing. He didn't eat anything. He said, well, I have stuff to eat as soon as I get into my office. If you get into your office. So from a teaching perspective, for many of you going into nursing and health science, when you get diabetics, feel free to review stuff like that. It's easy because sometimes they're very, very, we call it labile. The sugars go up and down. Pre-diabetics, they very often have a condition called reactive hypoglycemia. They eat something, it has a lot of carbohydrates, their insulin gets overproduced very, very rapidly. It's on the way to being not being. Diabetes, I know a lot. Moreover, if you're having diabetes questions in AP2, I am here as a resource. Not going to forget about you. Oh, Lord knows I can't. See, good. That was a good one. Look, I'm here. You need me, I'm here. So you pay me to be here. At best, you got me for another year and a half. Maybe a little longer, we'll see. You can note that upstairs. I'd like a couple more years. Moving on. So they're amitotic, high metabolic rate, all have the cell body and the processes. That's what we're covering today, the anatomy. Again, the perikaryon or soma or cell body, use the term nuclei differently. They make all the other stuff. And it's constant. They're very, very busy cells. That's why they need so much glucose for energy. That's the energy aspect. They use other materials as well. And very often they're pigmented. A lot of the neurotransmitters and other chemicals they make. Chromatophilic is a name for pigmentation. Chromatophilic means it likes color. The cell bodies are just one of them. That's how you pronounce that. And they're made in the rough endoplasmic reticulum. They've got a nucleolus. The plasma membrane is part of the receptive area that receives the input. So you're going to see and again, you're going to see a lot of areas when we're altering this membrane by whatever means, whether it's pressure, whether it's chemicals, whether it's light, all of these things change, and that's what sends the signal. We'll go into that a little bit about how the signal is transmitted, and particularly so at the very end, and I hope to get that done next week when we get the neurotransmitters in, because I think it's really important. Again, in the central nervous system, we call them nuclei. In the peripheral nervous system, clusters, we call them ganglia. If we go back and look at that, that is a sensory nerve. We're going to learn, and you don't have to write it down now, this is the unipolar area. This is a unipolar nerve cell. They're only sensory cells. These cluster right outside your spinal cord. When we look at the spinal cord, they're called dorsal root ganglia. Here's a clinical note from micro that we reinforce a lot in micro. You get a herpes virus, you can never get rid of it. It's with you forever. It becomes latent. Where it becomes latent is in these cells. Combination of those cell bodies are called dorsal root ganglia. They can be cranial nerves or spinal nerves. And then you don't have to worry about this because 1 or 3 out of you are going to get it anyway if you've been exposed to herpes virus, and nearly all of us have been. It's called shingles. We get into that when we talk about micro. Just in case you're wondering. And so that's really an important area about that with ganglia, and we stop here. We'll start talking about dendrites and axons and all this diagram as we move forward. Hasta mañana. Not hasta the day after mañana. Nice talking to you. And we're stopping.